What does TVR stand for? Well, it stands for Test and Vaccinate or Remove, and this is a good summary of what the project is all about. Within a control area, badgers were trapped, tested for TB using a set-side blood test, and then either euthanized and removed for further examination or released back into the environment. It was novel and exciting research, and it represented a significant government investment. It ran for 5 years in total, and it cost just over 4.5 million. It's important to understand at the outset, however, that it was a research project and not a pilot exercise or a scheme. Through the project, we gathered as much data as possible, and some of this data will feed into future research. And this is data such as body weight and condition of the animals captured or their state of lactation. We have a diverse audience today, and therefore the content will be intentionally science-light. We'll not be delving too deeply into the detailed findings. Our core audience today is our own frontline staff, and I would like to emphasise that. You are the people who are in daily contact with the so-called ordinary farmer, and you're vital in disseminating accurate information to the farming community at large. Some of you will already have had an opportunity to observe TVR directly or to participate in it, but others will not have had this opportunity, and this event is for you. So today will be all about what we did, how we did it, and what we found out. A TB strategy announcement is due in the near future. Therefore, policy implications for the project will not be discussed. That's something for another day. As far as how the meeting will run today, all of you who are listening in will find that your microphones are muted throughout the event, but written questions can be submitted for a Q&A session at the end of this morning. And it would be very helpful if you could direct your question to a specific speaker, if that's appropriate. There are a lot of people on the line this morning, so we may not be able to cover all your questions, but we'll try to pick out the key ones. Some of the research papers that you'll hear about have already been published, but others are still in draft. So I'd like you to respect the wishes of the presenters and not share further any data presented today which relates to papers not yet published. And finally, I would like to thank at the outset the speakers and the organisers for all their work in bringing this event together. And I'd like to also acknowledge the valuable contribution made by others who have retired or otherwise left the department. So without further ado, as the saying goes, I'll now hand over to Robert Huey, our CVO, and ask Robert to say a few words.
Thanks, Raymond. When you were talking about gremlins in the system, I thought you were talking about me, but never mind. We'll leave that to one side. I'm delighted that Raymond has taken on to organise this day. There's been a lot of external folks wanting to know about the details of the TVR project, and this is a good way of telling our own staff primarily first about what the results of the research project has been. As Raymond says, it's a research project, and I'd like to pay particular credit to Fraser. This wouldn't have happened without Fraser. And at the beginning of this all, there were a lot of people saying that it was a waste of money, that we shouldn't do it, that the science wasn't good. Lots of those things had some element of truth in them, but by persistence, we moved on with it. It proved to be the first wildlife intervention of any sort in Northern Ireland, which was important. And I feel that the work that we've got out of it, the ecology work, and the knowledge that we've gained from this research project is essential for the next step, which is a TB strategy, which I am hoping the Minister will announce in the coming weeks. Key to that will be our frontline staff. It'll be a new role, or an enhanced role, some might say instead. But it's important that everybody understands the science behind our TB programme and are able to talk to the farmers about it, because we must have a united message going out from the department and from our stakeholders. And we'll be working very hard to try and get this. We've got a one chance in a generation with this TB programme that's going to be announced to actually get on top of this disease and eradicate it. Yes, eradicate it. And I believe that if we do what we say we're going to do and do it properly, that we can succeed, but we have to do it all together. And this is the beginning of that process of making sure that everyone has the tools and the knowledge they need in order to talk to farmers and explain what we're doing and why we're doing it, and to help everyone, the farmers, yourselves, private practitioners, everyone involved in this programme, to move forward together to try and eradicate this disease. Not happen in my time, but perhaps within some of your working lifetimes, we will get rid of TB from the Northern Ireland cattle population. That's what this is about. I have to leave you now to go and do other stuff, but I hope to get back for the question time. Not that I'll be any use for the questions, but to support those who are. So enjoy the day, and I'll hand back to Raymond, who'll act as our master of ceremonies. Thank you.
Thank you, Robert. As I said at the outset, folks, we do have a busy agenda today. So without further ado, I'll hand over now to Dr. Fraser Menzies, who leads our veterinary epidemiology unit. And Fraser will present the first paper of the day, entitled Badger Intervention Options and Area Selection. Over to you, Fraser.
All right. Here goes this technology, so hopefully it will all work. Where are we? This one. Hopefully that should be my screen shared with everyone. Hopefully you can all hear and see this all right. So first of all, I'll just, as we said, it's quite a broad audience, so apologies to some of you that have heard all this before, but it's to give a bit of an outline as to why we took on TVR and the project outline and design, and then how we've taken that forward into choosing potential future badger intervention areas. So why TVR? Well, TVR is it's never been undertaken in the field. Hypothetically, it's been possible, but it's never been tried out. The most likely area, obviously, where you'd intervene with a TVR process would be in an area with high herd TB prevalence. Work from the south in the Greenfield study would suggest, if you select areas of high herd TB prevalence, then selecting for areas where there's high levels of TB in the badgers, it seems to act as a sentinel. The principle of the TVR approach seems to be sound in that if you selectively remove TB test positive badgers, that decreases infection load in an area, and those that are test negative, if you vaccinate them, then that's boosting the immunity of the remaining population, and hence the two act in a bit of synergy, which would hopefully accelerate the reduction in badger TB prevalence. Now, because we hadn't done this before, we did get our friends over the water, as it was then, FIRA, to do some mathematical modelling of the predictions, and it showed that TVR could have a positive effect on TB levels, provided no increase in badger movement, sometimes referred to as perturbation, which may lead to an increase in TB in cattle, the so-called perturbation effect, which was observed in England but not in the south. So as Raymond said, it's a 5-year project, but the modelling again would suggest that you first see the effect in badgers, and then there'll be a lag before you see any effect in cattle, and partly because the time step in badgers infecting cattle, and also you also have a lot of noise as far as cattle TB herd breakdowns go, because you've got potential other sources. But we did say we would monitor TB levels in badgers and cattle throughout the project. Yeah. So really, at the start of TVR, as kind of Robert alluded, we were all very naive, and set out these objectives. None of the diagnostic tests for badgers had ever been used in the field, and one of our main objectives is to evaluate this new Dpp test that had been launched. We're also, as I say, going to measure badger TB prevalence, and also important thing, because we had it in one area, was to monitor the badger ecological effects. Basically, most important being if removal of badgers increased movement of badgers within that area. and Maria will cover that later on. Also, as you saw, we're starting from a low base. That would help us gauge the kind of field logistics and the costs of such an intervention, and supply us with a lot of information from Northern Ireland region on TB transmission and data for economic modelling of TB intervention. At the start of TVR, I think we had about 2 staff that were kind of experienced at trapping badgers and vaccinate them. So a key step in this was build up our experience and core experience in our field staff in being able to carry out such interventions on a large area. And just acknowledge the many thanks to the patience of our colleagues down in the south, and also over in Worchester Park, and passing on their knowledge in these aspects to help us get off the ground. It was driven by a ministerial direction. We were told to do this in one area. So one area does have a few scientific limitations. We were restricted to before and after design. And the trouble with one area is that you're never sure if what you observe is happened by chance anyway, or whether it was an effect of what you actually did. So it does limit extrapolation of some of our findings. Shane's going to cover all the practical aspect, but just briefly, all trap badgers will be tested in the field. As I said, this is the Dpp test here. Set out here, it's a lateral flow device. You put the blood in one thing and see if any lines appear up at the top right hand corner here. First year was set as a control in the before and after design. So in the first year, all badgers were tested, vaccinated and released irrespective of the result of the test. It was also proved that it gave us an initial indication of how the test would perform. In years 2 to 5, badgers that proved test positive were euthanized and sent for post-mortem, and those that were test negative were vaccinated with a BCG vaccine. It's basically the same as used in humans, this badger BCG, as we call it, became licensed in 2010, so it was widely available. But there were problems with BCG supply over the course of the project, and we had to switch to BCG Sophia, which is another form of BCG then. AFB were heavily involved at the Veterinary Sciences Division, and we sent samples, duplicate blood samples for testing by the Dpp test and also by gamma interferon, and also various clinical swabs were taken for culture of M. bovis. These 3 tests were important in allowing us to evaluate the badger TB prevalence, and Liam will be covering that later on. Another part of the badger ecology, we were taking hair samples from the badgers to genetic profile them to get information on that, and our lab colleagues hopefully cover some of that. And then, as I say, the positive badgers were post-mortem, and there was bacteriological culture on them and from the clinical swabs. And this led to another subsidiary project on whole genomic sequencing of the M. bovis isolates. Again, this will be covered later. So when we're limited to one area, the badger ecological studies were probably more important in that we get a better indication from the results on these. And each badger, when it was first caught, was microchipped, so we could monitor its progress over the time of the project as to where we caught it and how many times it was vaccinated and tested. And the hair samples, again, as the badger, we had all the capture recapture sites, and I knew the genetics of these badgers as to where the relationships were. Another key component was that we put GPS collars on a subset of about 40 badgers each year to try and gauge if there's any increase in movements. These were done first year, give a kind of baseline, because we're only vaccinating them, we never removed any. So that provided a baseline of badger movements for that area compared to years 2, 3, and 4 when we removed badgers, and we can measure the movements against that. Maria will cover a lot of that in more detail. Our outputs, as I say, when we're limited to the one area, which is the Banbridge area, which is the bottom left on the diagram here, it does limit our sample size, so we weren't sure just how clear we'd be as to how we could say anything about TB, but we did monitor seroprevalence through the Dpp test. And as I said, by using the 3 tests that we used, we could calculate an annual TB badger prevalence. And then we also had the PM results and the badgers that were removed. We did do a power analysis at the start to see if we could measure cattle effect. These 3 other areas on this diagram show areas that were chosen as potential intervention areas or control areas if we were doing a randomized control trial. They weren't used, but we could use them as hands-off areas to compare the cattle TB prevalence. But when we looked at this, we needed to see a 6% reduction in herd breakdowns in the TVR area over the control areas to be able to say anything, which is a big ask on a 5-year project in such a small area. We had other metrics that we were looking at, but that's work in progress. Again, one of the big questions was, would TVR cause badger perturbation? So as I said, we had the microchip data and recapture of each where the badgers were captured, the hair samples and the GPS collars. All this will be covered later on, so I'll skip over that. Said we did some modelling earlier on to see what effect and maybe give some indication of the sample sizes we might need. And this showed that TB might be as effective as proactive culling, and the number of badgers to actually remove would be significantly fewer than you did use proactive culling. Again, this was the modelling suggested it might be dependent on whether there's a perturbation effect. But and just before TVR started, this paper by Weebly arrived, and as I've highlighted here, and Robert alluded to, there's question marks whether TVR would actually exacerbate the TB problem in the area. So there's a lot of unknowns at the start of the whole of this. So as part of the design process, we had to select these areas. As indicated, we had the 4 areas as potential intervention areas, and we based this on areas being at least 100 square kilometres with a high TB herd prevalence, high badger density and high cattle density. Now we've refined this process over the last few years to identify potential areas where we could maybe intervene in the future with the TB strategy as launched by TBSPG. Really we're looking for these characteristics, and this is not only this is coming from findings from the RBCT and the 4-area trial, where you're looking for a large area to have a maximum impact, and you also want to select an area where you can have prominent physical boundaries to stop the badgers moving in and out, and hence maybe diluting the effect of your intervention. And also, as far as cattle is concerned, we want to have areas where there's limited reintroduction of TB from cattle. I'm sure most of our staff already seen our what's called heat maps or kernel density maps, and we use these to try and identify areas of herds with high density of herds with TB reactors, and we took a 3-year period to try and stabilize these, because these so-called hotspots tend to move about a wee bit year on year, and I thought a 3-year period might be able to identify them. So we took this and we tried to identify areas where there was relatively large density of TB reactor herds, and when you select that out of the heat map, you get areas like this. The grey areas show these areas where there's a relatively high reactor herd density, and the figures in red show the actual areas in square kilometres. So that gives us our categories that might be interesting going in. Now, in 2008, Queen's University Belfast did a badger population survey on behalf of the Department of Agriculture, and this is what they produced. They found that about 34,000 badgers across Northern Ireland and about 7,600 social groups. And this population seemed to be stable. And the map here, the red areas show there's high densities, whereas the blue areas are where there's lower densities. And as you suspect, in the mountains and that, there's lower density compared to the good agricultural land is where you'll find most badgers. So from this, the Queen's developed a model to estimate the badger density across Northern Ireland. So this map shows the area split into 36 square kilometre hexagons. Again, the red areas show areas where there's high active main set density. Active main set really equates to a social group of badgers. So we have this, and we know that the average across Northern Ireland was 0.56 social groups per square kilometre, so just about 2 social groups per square kilometre. So we took this and we took an arbitrary cutoff of having areas where badger social group density was over 0.7. So it's above average, so we're trying to focus on areas where there's a relatively high badger population. And we mapped this, these hexagons on top of the previous high cattle TB dense areas. And where they overlap, we focused on them to try and identify areas. So taking the two together, this shows a map around about Omagh, where the red circle shows that the hotspot as outlined in the previously, and then what we tried to do was define that within the local physical attributes within the area. So within this area, to the southwest, we have the major road, the A5, which is a fairly good boundary, and to the west we've got Omagh town itself, and hills to the northwest and the southeast, and boggy land the northeast. So some of them are better boundaries than others, but you do what you can with the areas you're finding and try and define the boundary as you can. And most of the actual boundaries just map onto roads because they're easily identifiable and practical use in the field. The first two areas worked up in 2007 were the Coleraine and Omagh areas. As I said, I've highlighted some of the physical factors in the Omagh area previously. In the Coleraine area, up the east side here, we have the River Bann, which forms a major boundary, and the city of Coleraine to the top, and then some low-level hills to the west, whereas very ill-defined boundary to the south. So from this, once we've got the area defined, we can give some characterization to the areas as far as the area, the badger and cattle metrics, and the TB information we have relating to badgers and cattle in these areas. And once they're defined, the first step really is to carry out a badger set survey to try and identify where the badger sets are, which is very useful when you come to do any sort of intervention. And to date, as I say, we've got the Omagh and Coleraine areas, which we've already described, and been working on the St Field area over in County Down. So that's the areas we've worked up so far as potential intervention areas. So in summary, modelling outputs would suggest that TB can be effective in reducing TB in cattle levels. And our anticipated outputs, which are covered later in the talks, this is just really setting the scene for you. We want to demonstrate that selective culling using a real-time test in the field is a practical option. Evaluate the badger ranging behaviour caused by a TVR intervention, evaluating the Dpp test, and also monitoring the effect on badger and bovis TB prevalence. The study did provide a framework for other research studies, and we'll cover a wee bit of that in the future. and it was very useful in helping start off developing one method in which we have developed to identify potential badger intervention areas in the future. Again, that's work ongoing. There's areas we want to improve in it, but it's as far as we got. So that ends my presentation, and this is just a view of the mornings at sunrise from TVR. Now, this wasn't taken for me. I was, like most of you, still in your beds when all this was happening. But I'll hand over to Shane Collins, who I'll give you some of a practical insight into what was happening in the TVR as far as implementation goes. So thank you very much.
Thanks, Fraser. Hi, everyone. Morning. My name's Shane Collins. I'm a veterinary officer. I was based in Newry DVO, and I managed the field work on the TVR project for a number of years, and was also the ASPA project license holder. This morning, I'm going to run through some practical elements of TVR fieldwork. And some of you may have previous experience of this from field visits and prior training exercises, so there may be some familiarity out there. As this was a research project, it was subject to licensing under the Animal Scientific Procedures Act. DAERA is a licensed establishment under this act. The project was licensed, and all those conducting procedures such as blood sampling had personal licenses. All licensed procedures were carried out by vets. The work was subject to Department of Health inspector visits and annual animal welfare ethical review processes. Over the years, numerous refinements were made to the procedures and processes as required by the AWARE process. As badgers are a protected species, all individuals involved required licensing by the Environmental Agency to allow them to trap badgers between July and November, as outside of these times is the close period in Northern Ireland for any badger interventions. Badger welfare was of paramount importance to us throughout. All animals were subjected to clinical examination by our vets. We would treat sick or injured animals and did euthanize any animals thought to be suffering, for example from emaciation, although these cases were rare. All females were assessed for evidence of lactation, and those lactating badgers that tested positive were not removed. This again was a rare occurrence given the trapping commenced the first week of July each year when cubs in the main were generally weaned. So this is our area, 100 square kilometres. We had hard boundaries, the A1 to the west, the River Ban to the east, and the towns of Banbridge in the north and Merfilen to the south. As Fraser mentioned, to recap, AFB had conducted some preliminary badger set survey work on approximately 82% of the lands prior to commencement of the TVR project. The project was resourced with local veterinary service staff from the Newry Divisional Office and the surrounding DVOs. The advantage of this was that staff were known to the farmers and had already established relationships with them. This allowed us to gain access to approximately 94% of the landmass and aided cooperation during trapping regarding grazing patterns when machinery work was taking place, when we had our traps in place. So the farmers would work around us. Prior to commencement, staff were trained with DAFM, with Welsh Government and APHA in Worchester Park. This was the first time any of this type of work was conducted in Northern Ireland. Work took place in each area over 3 weeks. Each staff member was responsible for approximately 1 square kilometre during that 3-week time period. In week 1, permissions were sought and the land was surveyed for evidence of badger activity. Week 2, cages were dug in and pre-baited. And in week 3, trapping and sampling took place with cage removal at the end of the week for cleaning and disinfection. 3 teams of 6 inspectors worked with a staggered process so that one team was trapping every week. It meant therefore that the vets were sampling every week. And we had 4 vets sampling. Our highest capture numbers were usually on Monday morning. So you could have 4 vets working on a Monday morning, 3 on Tuesday morning, and maybe 2 on a Wednesday or Thursday. So this is our area. Work started down here outside Riff Island. First week of July, we started trapping. In initial year 1, this area here was used as a training area for all staff. And then over the course of 16 or 17 weeks, we moved seats up through the areas and finishing down here in mid to late October. The same areas were sampled the same week each year. At the beginning, there was a major procurement exercise. As you can imagine, there was quite a lot of equipment required. These are examples of some of the equipment we used. So then, week 1, it involved buying sets and checking for signs of badger activity. So around sets, that would be evidence of fresh soil heaps. Here we have evidence of bedding being left out from active set, digging in the area around it. The trees provided useful information as a sign of activity and also helped to identify boundaries between social groups. And then remote from the sets, runs were very useful signs of activity. We had quite a bit of a success trapping out on runs, and we didn't always concentrate our cages at sets, quite a bit of remote trapping. By the end of week 1, traps were dug in like so to allow badgers to get used to them in situ. And you can see here, this trap has been placed perpendicular to a run. So there'd be a run coming out here onto the fence. There was probably signs of recent activity such as badger hair on the barbed wire. All cages were individually numbered and their location was recorded. The labels also indicated that these were departments being used in a research project, and a phone number was also made available for anyone that had any badger welfare concerns. This is week 2. Ideally, the cages were pre-baited for 5 to 7 days before setting. Peanuts were the bait of choice, although we did try many others like jam, dog food, fresh, but we always reverted back to the peanuts. The stone with the bait underneath was gradually moved closer to the back of the cage. You can see here the stone back of the cage. Over the week to try and encourage the badgers to enter. Then in week 3, the cages were set for 4 days from Sunday night to Wednesday night and checked at first light the following morning. The trigger mechanism via a wire and string attached around the stone. You can see here the string comes up the back of the cage and up to the trigger mechanism. Badgers are pullers, and they test the peanuts, they would pull the stone back, triggering the cage. But we have done this over time, as we suspected some badgers were pushing the stone to the side. So we changed our method to using a string in the center of the cage to act as a tripwire. So this is what we want to find on a trapping morning. Here we have a nice fresh badger. No sign of clip marks or marker spray indicating that it had been previously caught that year. Recaptures a welfare assessment by a vet and had their microchip numbers recorded and capture location recorded and were released. Again, recapture data was useful in defining badger territories. Occasionally we did catch other species such as birds and foxes, rabbits, and these were also released after a welfare assessment. Animals were subjected to the following procedures as listed. I'll go through some of these now. So the anaesthesia, the veterinary officer estimates the weight of the animal and then injects anaesthetic intramuscularly. This consisted of ketamine, medetomidine and butorphanol in a 2:1:2 ratio. Dosage regimes were based on work from Worchester Park, which we refined in the course of the project so that we had an efficacious protocol. Animals were drained by a wicket. You can see here it's like plastic prongs in the cage. Occasionally we do in a cage and they would need separated to allow injection. I have a short video here, hopefully this works. I preferred to work quietly without any restraint. A lot of the times, if you worked quietly and calmly around animals, you could inject them yourself. They responded better to anaesthetic this way. They didn't get overstimulated or overexcited. And you can see there that that animal hardly moved while being injected. The vets then assessed the depth of anaesthesia and removed the badger from the cage when satisfactory. They were taken out and removed to a recovery box, this plywood box here. This doubled up as an examination table and then also was a box to place the animal to recover from anaesthetic after the procedures had taken place. There was a considerable amount of equipment needed to carry when vehicular access wasn't practical due to the location of the cages or the ground conditions. So the first thing we had to do was to get the blood samples taken to get the Dpp test up and running, as it takes 20 minutes to run. And jugular venipuncture was used. The amount of blood to be stipulated in the ASPA license. One green top for the field sample was taken, and then additional red and green tops to go to the lab. So the site's prepped, and then bled. The test was then conducted. This was done in heated, insulated boxes in vehicles, as the test could be conducted at room temperature. I would sometimes carry the back to the vehicles to be sampled, rather than carry all the equipment to the cage, and then after sampling, I would take the animal back to the cage to recover from anaesthetic. The animals were weighed so that the anaesthetic could be topped if required, or a reversal dose calculated. Then subjected to a clinical examination. All badgers were microchipped. We cured these extendable wands. It allowed us to read the microchip numbers while the badgers were in the cages. Oropharyngeal swabs and tracheal aspirates were taken. So this was a team effort. You can see here all samples were packaged in the field for dispatch to the lab. It wasn't always small like this. Sometimes this was done in adverse weather. Wound swabs were taken to be sent for culture. Parasite checks were undertaken. Samples of the parasites were taken for related Queen's University Masters projects. Here we are taking the length, neck, and neck circumference measurements. So there's a whole range of ecological data gathered during the process while the Dpp test was running. Also performed dentition checks as a rough guide to the age of the animal. And here we can see one of the vets, all the samples, they're barcoded and they're being uploaded via the digital device to our viewer so that the lab could see the data live. All data was recorded digitally. It was date and time stamped. This aided audit and compliance of licensing stations. It reduced errors, duplication of data. The lab had live data to plan for the resource required and the reagent required, and they knew when to expect the samples. And the manager also had a range of dashboards to be able to supervise remotely. So initially at the very start, it was a paper-based exercise, but this technology developed over time and it became well established over the course of the project. I've mentioned there some of the benefits of the live data capture. This was one of our dashboards. A range of data was easily accessible. We use this for our anaesthetic records to monitor the numbers of cages deployed and the location of where they were deployed. Here we have each individual badger microchip number and all the lab samples recorded against that animal, and then all of the clinical examination results were recorded as well. So cages were checked. This is early first thing in the morning. Data was inputted into the Trimble devices, and the vets would then know that this animal needed sampled. This shows our sampling locations in a given year. You can see here that there's a concentration of badgers up in the northeastern boundary. This was an area of high badger density due to an old railway line. There was an embankment running down along here, and then also as a boundary we had the River Bann, and there was a squeeze of badgers against that hard boundary. This shows sampling location right down to the individual field level. So you can see here we're right up against our boundary. We can see cage location, cage numbers, and then where badgers were sampled. And you can see that the badgers were generally sampled beside the cages where they were caught. This view here again has our sampling locations. The orange squares are the cage locations, and then the red and green, that's our permissions. So we had access to 94% of the lands, but we did have some blocks of land in the centre where we didn't have access to, although it didn't hinder us too much. We just placed our cages around the boundaries when there were signs of activity. So these are our Dpp tests. You can see this is in an insulated box on top of a heat pad. This test has a weak line 1 positive, and any colour change on line 1 or line 2 was deemed positive. Here we have a stronger line 1 here. Positive animals, they were euthanized by lethal injection while still under anaesthesia. If necessary, if the animals were coming round, we would top up the anaesthesia before euthanizing them. The animals were packaged as per ADR regulations, paperwork was attached, and the carcasses were dispatched to the lab straight from the field. We had a designated ADR driver and van on site every morning, and the samples were also delivered to the lab in this way. Test negative badgers, they were vaccinated intramuscularly with Badger BCG, that was a licensed product, or the Intervax BCG Sophia at the latter stages of the project, as supply of the licensed BCG Danish vaccine became an issue during the project. We also did some additional sampling to assess the effect of BCG Sophia vaccine on the Dpp test, and Emily will discuss that later. If suitable, the appropriate weight neck circumference within an appropriate area and within a social group that we were targeting. Some badgers would be fitted with a GPS collar. Again, Maria will mention more about this in the next talk. Animals were allowed to recover in the recovery box, or if safe, beside the cage. Most were recovering by the time procedures were completed. Over the course of the project, I moved away from reversing the sedation as they had a smoother recovery when they were allowed to come round from the anaesthetic themselves. On occasion, we would allow them to recover in the cage. We would place them back into the cage and come back and release them and supervise them if needed, crossing roads, or if there was embankments or water courses nearby, we would allow them come round fully in the cage and then come back and supervise the release. You can note here on this animal the clip mark and stock mark to the side. That's how we recognise the following morning whether this animal had been trapped recently, either the day before or maybe in a previous week. So the range of samples were submitted to the lab. Fraser mentioned these previously. And then after sampling, after leaving every field, all equipment and vehicles were thoroughly disinfected between sites. All cages were removed at the end of trapping week and brought to a road service depot within the area for power washing and disinfection before redeployment in the next area. Every effort was made to separate cattle from the cages via electric fences. This prevented cattle triggering cages and also any potential nose-to-nose contact with badgers in the cages. These were our capture and removal numbers over the 5 years. You can see we had quite high numbers of recaptures, which was encouraging for us, as it indicated that badgers were not put off by their experiences in the cages. You will note, especially in year 5, higher recapture numbers. Year 5 was 2018, and if you remember, it was a very hot, dry summer. There was a scarcity of natural food sources, and as a consequence, recapture numbers increased. So to conclude, we had a wide support of stakeholders, which was conveyed at many field visits by interested groups. The welfare of badgers was very important to us. We achieved our field objective of supplying the information and data to inform on future bovine TB strategies, and we built up a bank of field experience among our staff. It must though be noted that a TVR approach is costly and can be quite resource intensive. And finally, I would like to thank all the staff involved in the project and all those farmers who allowed us onto their lands. I'm happy to address any questions in the Q&A session, or I think there will be an email address through the wildlife unit that any further questions can be sent. So that's me. Thanks very much.
Okay, thanks for that, Shane. We'll now hand over to Maria, who'll present the next talk on badger ecology. Thanks, Maria.
Good morning, everybody. I hope everybody can see my slides. My name is Maria O'Hagan, and I work for DR's EPI unit. The talk that I present to you today will be focusing on the badger ecology. And I'll give you an overview of the ecological parameters that were collected during the TVR project. So this is an overview of the talk I'm going to present to you. First of all, we're going to have a look at the data relating to the badger sets that were collected. And from there, also, I'm going to give an estimate of the badger population within the TVR area. I will give you an overview of all the number of badgers captured and removed, and talk a little bit about cage-related injuries as well. The last part of the talk and the most extensive part will be focusing on badger ranging behaviour. So the first slide then is in relation to the badger. So as you know, and as mentioned before, an extensive set survey was our project. That started even before the real project had started, after we did some set surveys as well. The table on this slide will give you an overview of how many sets were found in the different locations. So we had the hedgerows, open farmland, scrubland and woodland. Most of the sets, 85% to be precise, were located in hedgerows, and this is consistent with previous studies that were conducted. Main sets, in which we were particularly interested in, because they are assumed to be a representation of a social group, accounted for about 11% of the total sets identified. The majority of sets were outlier sets, 64%, and compared to previous conducted studies like the TB biosecurity study, that is a very similar figure. So in relation to the sets, we looked at the mean distance between the main sets. Part of the reason was because we were mainly interested in that, because it is a measure of creating social grouping as well. So the average distance between main active sets in the TVR study area was 820 meters, so just shy of a kilometre. And the map on this slide shows the locations of the active main sets found. You can see, as Shane mentioned before as well, there was a slightly higher density on the northeastern side of the study in badgers and in sets as well. So the number of main sets that was found in the TVR area closely matched those predicted by previous model conducted by Queen's University Belfast, that was mentioned by Fraser. In order to double check that the assumption that an active main set equated to one badger social group, there were motion activated cameras deployed as well. And results from that indicated that equating active main sets to social groups slightly overestimated the number of badger social groups. Moving on then to the estimation of the size of the badger population in the TVR area. Now, this is notoriously difficult, which might not be surprising, because obviously over that 5-year period, the badger population is not static. And that is because there's badger migration in and out of the area. Badgers are born, badgers die. There's always a yearly or natural variation in population. And then on top of that, to actually make the measurements, I suppose, harder, badgers are nocturnal as well. So a number of methods based on capture-recapture figures, along with results from previous modelling by Queen's University Belfast, equated to a population estimate of about 550 individual badgers in the TVR area. So seeing that the TVR area was about 100 kilometres squared, that equated to about 5.5 badgers per kilometre squared. There was a total of 80 social groups identified. So that equates to about 0.8 social groups per square kilometre. And that equates again to 4.4 badgers per social group. So the number of badgers captured and removed then, Shane already spoke about that. This slide gives an overview of the number of badgers captured. So 271 to 341 unique badgers were caught per year. And over the 5-year, 1,520 unique badgers were captured. Of those, 11 to 56 badgers were removed each year, with no badgers being removed in the first year, as previously said. So based on these figures and the percentage captured badgers removed based on a population estimate of 550 badgers. That varied then from 2 to 10.2% of the estimated population removed per year. In total, 108 test positive badgers were removed from the area and 1,412 vaccinations were administered. If we break that down, then the captures by sex of the badger and weight. So the majority of badgers that were captured during the TVR project were adults, 72% to be precise. And unsurprisingly, the average weight of male badgers was higher than female badgers. So that was 9.9 kilograms versus 9.3 kilograms. The majority of captured badgers had a good to very good body condition, and this body weight increased consistently from July to October. Then we looked specifically at cage-related injury as well, and that was a very positive story because there was no cage-related injuries in 97% of the trapped badgers, which is obviously very high. Very few badgers had any cage-related injuries at all, and only 44 out of the 1,520 unique captures had an injury recorded, and all of those were considered to be very minor, as you can see in the table below. 2 badgers had neck injuries relating to GPS collars, and 1 badger had a long-standing broken jaw, so that last one was more of an incidental finding. Moving on then to parasites, they were very commonly observed, as you can see from the table below. So we looked at lice, fleas, and ticks, and as I say, they were very often observed. Of the ectoparasites, lice were the most commonly observed, with 83% of badgers having them, and ticks being the least commonly observed, with only 29% of badgers having them. As Fraser already mentioned before, a subset of badgers in our area received a GPS, or a global positioning system collar, and suitable animals were selected for that based on their weight. So the animal had to be at least 8 kilograms in order to receive a collar, and that's consistent with what other studies have done as well. Now, the aim was to deploy about 40 GPS collars per year, so 40 different badgers, and preferably they would have been put on 2 badgers per social group, preferably a male and a female, and again, preferably during each of the 4 years. The reason I'm saying 4 years was 2018, there were no GPS collars deployed, as that year was used for retrieving collars that might have still been on badgers. So for 4 years, those collars were deployed. Data were recorded on a daily basis, scheduling 8 locations per night at hourly intervals from 9 o'clock at night till 4 o'clock. So this table then gives an overview of the badgers collared each year. So there were about 35 to 41 badgers collared each year, which is very close to our aim, which was about 40. So that was well achieved. And there was slightly more males than females collared. Per badgers, a median number of 104 to 176 days of data were collected, which equated to 724 to 909 data points per badger per year. So that's a vast, vast amount of data. All those data then, they were plotted on a map zone. This map's starting to look familiar to you now. Basically, what you see here is this picture on the slide is an example of what that looked like for the year 2015. So every badger had his own collar. and you can see the dispersal of the badgers involved within that year. So the main reason why those GPS colours were allocated was to look into ranging behaviour. So all those GPS data points that I just spoke about, they were plotted on the map, and for every badger, a home range was calculated. So a home range is really the badger's territory, and it's the area that the badger usually resides in. In this case, what we did is we calculated home ranges based on the top 95% of locations that the badger mostly resided in. The reason why we did that was it has the advantage of getting rid of any outlier values, so that you really take in some sort of a core of where this badger resides. So these calculations were performed for every month for every badger that was coloured. And an example of what this looks like is portrayed on the slide there below. So you can see that all the little data points plotted, so the little black dots really, and 95% of the area where the badger resides is then encompassed by a polygon, which in this case is the purple area that you see on the screen. So one of the main reasons for calculating these home ranges over time was to see if the badger intervention actually had an influence on badger ranging behaviour, especially to see if they started to roam any further. So this refers, as you've probably all heard about, is the well-known perturbation effect. So this table looks at the initial results, and looking at these monthly home ranges by year, you can see that there's really very little difference between the years. The home ranges vary between years from 0.89 to 1.08 kilometres square. Just to reiterate again that in year 1, no intervention took place. So that's really like a baseline figure, whereas in year 2 and 4, intervention did take place. However, this is just a descriptive figure. It was important as well to take other factors such as the sex of the badger and the month of collection, for example, that they were taken forward into the further analysis. So that was done. Those factors were taken into account, and year was taken as a proxy to see if intervention had effect. So remember that I said before that the first year of the project, 2014, no intervention took place. And if the intervention had had an effect, we would have seen a change in home range sizes in the years 2015 to 2017 compared to the first year. However, at multivariable analysis, so the analysis when all the factors were taken into account, the home range sizes did not significantly differ between years. So in that assumption, we could say that an important finding is that we have no evidence based on this study that TVR intervention causes significant change in badger roaming behaviour. If we look at that in a little bit more context, we can see as well, we can note that really only a small proportion of badgers, which was 2 to 10.2%, as mentioned previously, was removed annually compared to the majority of badgers in studies based on proactive calls. And furthermore, as well, what we can see is that more badgers were actually removed from the TVR area by road traffic accident fatalities than through the intervention themselves. So to put a figure to that, for example, in 2016, 29 road traffic accident badgers were reported in the TVR area, while only 11 badgers were removed through intervention. So that puts it into context as well. Furthermore, we looked at the distance travelled, not so much specifically over the years, but just in general to get some more grip from an ecological point of view. So we looked at the dispersal movement of badgers calculated by the distance moved from the centre of their home range. And within the TVR project, the most commonly maximum distance move from the centre of a home range was only between 1 and 2 kilometres, so they stayed fairly close to their centre. We also found that 75% of badgers did not attempt to go any further away than 3 kilometres from the centre of their home range. And that's reflected again as well in the proportion of locations measured more than 1 kilometre from the centre point of the home range, which was only 6.1%. Looking then at the differences between males and females, males tended to have long distance movements more frequently than female badgers. However, if we looked at it a bit more details, females made long distance dispersal attempts that were further away from the centre of their home range more often compared to I have to say that again. So females made long distance dispersal attempts that were further away from the centre of the home range compared to males. So the last bit I'm going to talk about was just a smaller project, and the main question that we aimed to answer with that was, do badgers cross a main road? So the reason why we asked this question is because badger intervention areas are often chosen because they have natural boundaries, as mentioned before by Fraser and his talk. And in the TVR area, one of these boundaries was a main road, which is often the case for badger intervention areas, that roads are chosen as some sort of a borderline. So we wanted to see whether this main road really worked as a deterrent to keep badgers from moving in and out of the intervention area. You can imagine that that's something that you don't want, is that there's constant movement of animals in and out of the area, because that would be very difficult then, in relation to results that you extract from your study. So to answer this question, 5 badgers in the western part of the TVR study area were specifically GPS coloured in 2017, and to see whether they would cross a major road. Now, the major road in question is the one you see on the picture on the slide, and it was the A1, which is a dual carriageway that goes from Belfast to Newry via Boundbridge. So as I said before, this road acted as a boundary for the TVR story area, and with the main aim of discouraging or preventing movements of badgers in and out of this area. So the length of this border was 9.4 kilometres. In order to answer that question, 5 badgers that were trapped in the TVR zone close to the A1 were fitted with a GPS collar in October 2017. So those GPS collars were scheduled to send out 24 locations per night at 20-minute intervals from 7:20 at night until 3 o'clock at night. So those locations came through much more frequently than the collars employed to the other badgers in the zone. And the reason we did that was to get a more precise track of their movements. So the table on the slide gives you a bit of an overview of the badgers that were involved in this part of the study. So the results then were that 4,313 location points were collected or recorded in that manner, and only 2 of those were recorded on the western side of the A1. And as you can see from the picture fairly clearly, that was just one badger that crossed over, resided there very, very shortly, and immediately moved back again. Other badgers, though, as you can see from the pictures on this slide, moved very, very close to the A1, but didn't seem to cross it. You can really literally see the badger pushing up nearly against the road. However, to put this into perspective, we can look at the RTA badger survey, which has been running since 1998, and 4 badgers that were collected from the A1 section of the TVR zone since this survey started in 1999, 1 in 2000, 1 in 2015, and 1 in 2018. However, that's likely to be an underrepresentation because often badgers that are killed on large roads, main roads like that, might not be collected due to it being dangerous or the badger being quickly damaged because of heavy traffic. However, even though it was a very small study, it does provide some evidence that the A1 provided a good barrier for badger movements, which is important to know for future badger intervention products. Again, it was small, but we find that these findings are supported by evidence from other studies as well. So that comes to the end of my talk. Just a recap then of the main points. And as I say, this is only a very brief overview. So the majority of sets were outliers and they were located in hedgerows. The population estimate was 550 badgers in the TVR area, distributed over 80 social groups, equating to about 4.4 badgers per social group. 271 to 341 unique badgers were caught per year, and the percentage captured badgers removed varied from 2 to 10.2%. Very few heat-related injuries were found, and ectoparasites were very commonly observed. Home range sizes were not significantly altered by badger intervention removal that was applied in the area, and that's one of the main findings, I suppose, in relation to ecology. We found as well that 75% of badgers did not go further than 3 kilometers away from the center of their home range, and that in general, badgers rarely crossed major roads. As I say, it's a very brief overview, and I would like to thank you very much for your attention, and many thanks for all the people involved in the study, and thank you very much to Catherine as well for providing the photographs that were used for the slides. Short break will commence now.
Thank you, Maria. Maybe just before we break for a short time, because our speakers have all been so well behaved and we're a little bit ahead of time, we'd maybe just slip in a couple of questions now before we break. The first question we have here is one for Fraser. Fraser, could you explain what perturbation means and what the difference is between perturbation and a perturbation effect?
Yes, apologies. I kind of rushed through my talk there, and for those not familiar with terms, perturbation is really referred to as the increase in badger movement over what is normal observed. So badgers are a very hierarchical creature and they're very territorial. So they tend to have very distinct territories, as maybe outlined by examples in Maria's talk. So when the population is stable, they very much stay within their territories and zoom off now and again for a night out. But when you start removing badgers from an area, it upsets the social structure of each social group, and the badgers might start increasing their ranging behaviour. Now, when they start increasing the ranging behaviour, you're increasing their contacts, and so there's increased badger movement and contacts. Well, the increased movement is the perturbation. When you start increasing the contacts, both with badgers and with cattle in other areas, when they increase their ranging, the increased contacts can result in increased transmission of TB from such badgers. And in the randomized badger control trial in England, what they found was that when they did proactive culling in an area, it decreased the TB levels in cattle herds in those areas, but it increased TB levels more or less by the same amount in a 2 meter buffer outside that area. And the hypothesis for this was a TB perturbation effect, which is those badgers were disturbed because of this intervention, and they increased their ranging behaviour, increasing their contacts and hence transmission of TB, and causing this deleterious effect in the surrounding areas. Now, it was seen in, briefly mentioned, it was seen in England in their trials, but not down south. But part of the reason for not seeing it down south has been given as, if you choose areas with good physical geographical boundaries, you've got quite a close population, and the 4-area trial down south had some of these more clear barriers, and also the social group size is smaller there, so there's a whole lot of factors come into this kind of perturbation and perturbation effect. So I hope that explains it.
Thanks very much, Fraser. That's very useful. Any of you out there that have had any interest in the cattle and badger issue at all will have seen these terms referred to in various articles. So it's good to have them clarified for us. We'll maybe just slip in one more question before we break, and this time it's a question for Shane. And the question is, how many badgers could a single vet process in a morning, and would it make any difference if they were doing this work in a live intervention as opposed to as part of a research project, as was the case here?
Yeah, thanks, Raymond. That's actually a good question. And the answer really is it depends. As with all field work, we know every situation is different. Some mornings I could sample 7 or 8 badgers, and other mornings maybe only 2 or 3 in the same time. Now that's down to the location, getting access to these animals, how they respond to the anaesthetic. We were bound by licence. The licensing stipulations were the badgers should be processed as close to first light as practically possible. This was to avoid them being exposed to daylight and the elements, and if they were lactating females, they would be away from their cubs. So the goal each morning was to have field work completed mid-morning after starting at first light. So the rate limiting factor here really is the Dpp test. It takes 20 minutes for the Dpp test to run. So between an intervention and the study, in an intervention we may not be taking all the additional samples and the measurements and the clinical examinations, but you still have to wait for that 20 minutes. So as a rough approximation, I would say 30 minutes per badger. On occasion, I could have been a lot quicker to that if we were at a set and there happened to be 3 or 4 badgers in cages at a set. You could knock them all out at the one time, had the test all running at the one time, and you could get them quicker that way. Or otherwise, you could have 3 or 4 spread out all through the 6-kilometre area that you may have to walk 15 or 20 minutes to get to. So it's back to what I said at the start. It all depends. And the one thing I would say with the Dpp test, you have to wait 20 minutes before you can call it negative, before you go to vaccinate the animal. But if it was going to go positive, it usually goes positive quite quick. So you do have an indication if an animal is going to be positive before the 20 minutes, so you can plan accordingly. So I hope that was helpful and answers.
Yes, thanks, Shane. That's very interesting insight into what you found. Right, I'd like to thank the speakers that we've heard from so far, and also to Shane and Fraser for taking those couple of questions. And I hope that you're finding the presentations interesting and useful, and that you're getting a bit of a feel for what TVR was all about. I'd just like to remind you all that questions can be submitted through the Q&A function, and we have questions coming in. We may not be able to deal with all those today, but we are noting them carefully to address at some point later. If there's a particular person to whom you'd like your question addressed, then let us know, and we'll try to deal with that as well. As I say, our speakers have all been very well behaved, and we're a little bit ahead of time. So we'll have our comfort break now, and then resume again at 10 past 11 rather than 20 past 11 as stated in the agenda. Thank you. Raymond,
Would it not be better waiting until 20 past, because some folk might be joining then, specifically for the talks? So they might miss half, so we can have an extra long comfort break maybe.
Yes, that's fine. Yes, that's fine. We'll start again at 20 past 11 then. Okay, thank you.
And you thought you were going to get more time to talk, Robin, did you? Rushing through this morning.
No, that's fine. No, that works. I think that works okay.
It's a bit difficult because you don't engage with the audience, so you're not sure, you know.
Yeah, I see there's 96 participants.
It was over 100 for a while, but we'll see if they come back over coffee or whether we get them off.
Yeah. Yeah, total numbers were over 100, so we still have 85 attendees on. So just to be conscious of the fact that everybody's still on as well, even though we can't hear them. So
is that Carl? Yeah.
Yes.
Carl, yeah, I'm not. Well, screen share isn't highlighted yet. Obviously, sure it's not.
Yeah. No,
that's okay.
Well, I'll hand over to you once Raymond.
Raymond's done. That's grand. Yeah. Okay.
And if you let me know if you want to switch to Adrian, if you just say you're going, Adrian's going to present, and I'll switch to him. Okay.
Okay. Yeah, I think it's slide. I can't remember what slide number it was, but it's on the slide.
Okay.
I would say the weather's helping us today.
The lockdown is helping as well.
Sunshine in Bangor. Liam.
Is it? It's lashing here. So was it working from home or living at work?
Oh, no.
Well, I can't even get the dog to run safe. That's
very interesting session, Raymond.
Thanks, Robin, and welcome back, everybody. According to my laptop, we're just on the nose of 20 minutes past 11. So I hope you all enjoyed your break and you're coming back to us refreshed and ready for our second group of talks. And we'll have a short question and answer session at the end. So we'll lead off this session with a talk jointly presented by Adrian Allen and Robin Skuse from the Agri-Food and Biosciences Institute, or AFBI as we know them. Their talk is entitled Current Lab Research Activities.
Okay. Okay, good morning, everybody. Hopefully you can see the screen share and you can hear me okay. I'm Robin Skuse. I'm a veterinary research officer in AFBI, as Raymond has just said, and my colleague who I'll pass on to in a few slides is Adrian Allen, who's a senior scientist and both in veterinary sciences. AFBI is DAERA's non-departmental public body science provider. So what I just wanted to say thanks for the invitation to present this work today, and it's really the outputs of a fully governed DAERA research project, DAERA 15307. And we're going to, between us, talk about investigating TB transmission dynamics using genome epidemiology. And this is a project linked to the TVR project that you've heard about this morning. And I just wanted to set the scene a little bit. Taking it right back to the TVR system and TB in general is a multi-host episystem with a notoriously complex epidemiology involving within-host transmissions in cattle, within-host transmissions in badgers, and some level of between-host transmissions directly or indirectly through potentially affected environment. Now, we think the model is likely correct, but we've been dealing with this disease for decades, but there's still some surprisingly big unknowns. And as you've seen, and there's a lot of similarities with the coronavirus pandemic, association in epidemiology isn't necessarily causation. And there's still debate and evidence coming in on the importance of direct versus indirect transmission in this system. The current evidence, and you may hear some more about that this later, the current evidence using camera traps, cage traps, and proximity collars on cattle and badgers supports a role for indirect transmission, possibly some component coming through a contaminated environment, which needs further research. Maybe stating the less than obvious that epidemics are largely unobserved, and that's a function of moderate sensitivity of diagnostic tests, and in a wildlife setting, difficulty of trapping a representative sample of your wildlife. So you only ever really see the tip of the iceberg, and you're dealing with the transmission dynamics at that scale. So there's been a lot of debate and a lot of interest in what's the relative importance of the transmission routes between cattle, between hosts, and within hosts. We're going to try and address that today. So really, we're trying to use the genetics that we understand of the pathogen to try to understand and exploit genetic variation in the bacteria that cause TB in humans and in a wide range of different animals. These are the TB bacteria where they sit with other mycobacteria. But this group here, the tuberculosis complex, infects a wide range of different hosts. But this whole group of organisms they're exceptionally genetically homogeneous. That whole group is 99.95% the same, even at the genome sequence level. So until fairly recently, we were assuming that all TB strains and isolates were the same. But now that we have access to the whole genome sequence for the human pathogen and the bovine pathogen, we now have a toolbox which provides a hierarchy of different tools, in this case genetic markers, that inform on different evolutionary and geographic scales. So you'll hear us talking about global resolution, regional, local resolution, and sort of microbial forensics. And the book analogy is quite good here. The current strain typing techniques that we've developed are looking at just a number of words in a book, if you like. If the book is the whole circular genome of TB, we're just looking at variation in a small number of words to try to understand the difference between books. But the whole genome allows us to read all the letters in the manual and make those sort of high resolution comparisons now. So we have a number of genetic tests that resolve at lineage, clone, strain. I'm going to just talk about some strain level resolution, but the ultimate resolution is the whole genome sequence type, which you'll hear about from my colleague Adrian. So I'm just going to talk about TB strains at the strain level, and this is the technique that we've actually developed from first principles. So when we apply it to the Northern Ireland population, sampling cattle herds at herd level, we see this very striking picture of the distribution of the different strains and different colors here. So these are the 10 most prevalent of about 300 different strains that we've detected in cattle and other animals in Northern Ireland. And it's a herd level sample. So, I mean, like the COVID stuff, consider these tests are useful for track and trace. So this herd level strain typing shows this striking pathogen genetic structure in the population and this very strong geographical localization. The ecologists would call this anchoring. So something is keeping TB in the area, and then there's this phenomenon of clonal expansion. It expands out from the position that it started. So we're asking, why does the epidemic look like this? Look as structured as this in the pathogen, given what we think we know about cattle movements. This is a movement network by AFBI colleagues, which is remarkably stable, but it shows the cattle movement, the extent of it, and it doesn't easily explain this structured picture. And again, this is cattle recorded transmissions between and within the various divisional veterinary offices. So these are the tools that we have, and I just want to step you through some of the observations from applying these tools in a small area. So for TB strains, as you've heard from Maria, she talked about badger home ranges. We can identify risk surfaces and home ranges for these pathogen strains as well. And I'll just show you what that looks like. TVR is the green area. So it's obviously sitting right on a strain 6 home range or hotspot. So strain 6. And strain 297, which is a variant of strain 6, is again in the area, as is strain 122. So that's kind of what seems to be prevalent there. But just looking at herd level, cattle data, this is just looking at the confirmed cases that confirm the outbreak in the cattle herd. They're split about 50-50 between purchased animals and animals that have a homebred ear tag. But the purchase of animals is associated with an increasing diversity of TB strains. This panel here, this is strain down the side, year across the top, and this is all animals, purchased or homebred. If we strip out purchased animals and just look at the homebred animals, strain by year, it's much less diverse. So limiting to homebred reduces the strain type diversity. So movement is bringing in diversity from other regions. And just looking again at cattle herds, but this time within herd at animal level, the strain types. So cattle herds can have multiple types per year. These are TVR anonymized herds. Herd 11 there has 6 different strains in 11 isolates over this in a given year, which was 2015. But we also see evidence consistent with, if you see it here, strain 6. There's a big outbreak there, 15 of the same thing, 21 of 297, 17 of 6. So you get out transmission and big outbreaks. So the evidence is consistent with multiple sources putting in the diversity, but within herd amplification, we've got evidence of it. You can see it. It's very hard to quantify, but you have got that within herd amplification effect, which is clearly seen with the strains like 6 that we would consider endemic to the TVR area. So moving quickly on and just doing a very simple Venn diagram, looking at the sharing of TB strains between TVR cattle herds and badgers. There's a big group of strains at pretty low frequency that are cattle that are only isolated from cattle. There are 2 here which we only isolated from badgers, but we have previously isolated these in our cattle surveillance from other regions. And then we have this group of strains that are shared. For example, 6 at fairly decent amounts and 297 at fairly decent similar amounts. So what we're saying is that it's clearly a shared epidemic. This doesn't tell us anything about direction, but we'll come to that. So this is kind of where we got to before at the start of the DARA project. So we can say that cattle and badger TB strains resolved at the strain level. MLVA types, we make these data available to you for outbreak investigations. So the strains are in badgers and cattle are associated, they're co-localized at this regional scale. So the badger strains of the same strain are shown in stars here. I hope you can see it if your eyes adjust, and the cattle herds are the same colored circles. So there is this co-localized sharing, and a statistical analysis, nearest neighbor analysis, shows that hosts with the same strain type tend to be closer. So cattle to cattle of the same strain are closer than cattle to cattle with a different strain. Badgers and badgers of the same strain are closer to each other than badgers with different strains. Cattle and badgers that share the same strain are closer than those that don't. This particular tool, it gives you this visual impression of a shared epidemic, but it lacks discrimination for microbial forensics. It lacks, we've been lacking direct genetic evidence of transmission chains that link cattle and badgers at the farm scale where it matters. We can't say anything about directionality from the strain data. So can genomic epidemiology help? And I'm going to pass on, hopefully my colleague Adrian can share screen from here. And hopefully, okay, you. I'm going to mute out.
Okay, can everyone see that okay? So yeah, as Robin said, the strain typing is an extremely useful tool, and it gets us to the point where we can actually localize microepidemics of infection across the landscape, and we see that those cluster a lot. And the directionality question, unfortunately, using that technology is impossible purely because we lack resolution. I mean, if you zoom in on one of these clusters, say this green one here in South County Down, if everything looks the same, it has the same strain type, we can't figure out, who's infecting whom in that particular scenario. So the idea has been to try and improve the resolution of that. And one of the ways we can do that is by using genome sequencing. Why is that useful? Well, back to kind of what Robin said, it's because with this book analogy, and instead of just being able to look at 8 pieces of genetic information in the M. bovis genome, we can actually look at all of them, and there are 4.3 million sites of genetic information in the M. bovis genome. And once we know that, we can actually we can look across multiple isolates of M. bovis that we get from cattle and badgers, and we can find mutations there. And our chance of finding mutations when we look at everything is greatly increased. And so like a typical mutation, say this is a typical DNA sequence within the M. bovis genome, and you find maybe the first T has changed to an A. We call this a TA, SNP or TA mutation. And when we look at multiple isolates, we're going to find thousands of those types of mutation. And once we have those thousands of those mutations across multiple samples, we can actually start building a family tree, if you like, of the bacterium. And these family trees, they're really, really similar to what you would have seen for like genealogical studies in humans. For instance, back here we have the ancestor, the granddaddy of everything, and then moving forward in time, we get to other ancestors, later ones, and then at the tips, we have the samples that we have taken and genome sequenced. So what we call these internal nodes, because these are ancestors that we didn't sample. And also the horizontal branch lengths that you see here are the number of mutations difference between isolates of M. bovis. So when you count up the number of mutations, you can see here that badger here, isolate from this badger and the isolate from this cow are much more closely related than say this cow M. bovis and this badger M. bovis, because they're a greater distance away from each other on the tree. So things that are closely related cluster together on the same branch like these 2 isolates. So that was the general idea. That's why we want to use this technology, because it takes the existing strain type information that looks homogeneous, where everything looks the same. And if we apply genome sequencing, we can explode those clusters up into much more refined and information about relatedness and build a family tree within a cluster. So our first foray into this was back in 2012 with University of Glasgow, with collaborators that we're working with there. And we wanted to do just a proof of concept study by targeting one strain. The strain we chose is one that had relatively recently emerged called strain type 10. So it's this, if you look at the map here, it's this little blue one just on the western shores of Strangford Lough. So anything that's a circle here are cattle, anything which is star are badgers. So they share the same strain. So it was worth looking at this and applying genome sequencing. And our colleagues in Glasgow, we did all the culture work, we did all the extraction of DNA. And then they did the whole genome sequencing, the high performance computing and the mathematical modeling, which allowed them to build this family tree over here. And once we had that information, we could build what was called a time-stamped family tree, or phylogeny, which gave us the ability to actually investigate the dynamics of what's going on here. And the thing that really became apparent was that within these different herds of cattle, the cattle isolates and also badger isolates were extremely closely related, particularly badgers from which were associated with herd 3, and then herd 3 itself was closely associated with animals from herd 5. So what we can say from this information, just by eyeballing the tree, is that this is a shared epidemic, and there is recent evidence of transmission between cattle and badgers in the area. And this has actually been cited as probably the first direct evidence of ongoing transmission between cattle and badgers at an individual farm level. There were also some other things that fell out of the analysis, which would suggest that cattle-to-cattle spread or amplification looked to possibly be quite common in the model. So you could have had the case where animals were left behind after skipping the skin test, which then reinfected other cattle in the herd. So you had this amplification effect within herd. Hannah, who did this work as well, also found that recorded cattle moves did not seem to explain the kind of dynamics in the area. So this sort of technology really did improve. It gave us a real chance to pilot it, and it showed us that we can actually improve our knowledge of the disease using this method. We couldn't do directionality from this for a couple of good reasons. The first one was that we didn't have the exact mathematical tools to do directionality work. That's changed. I'll come back to that. The second was that our sampling in this area, it being just a pilot area, was not particularly systematic. And that's particularly on the badger side. We were relying on badgers which were captured as part of road traffic accidents. So a much more systematic sampling would be a better way of doing this, to actually go out and properly sample the badger population and not just rely on the random chance of picking up ones from the same area with the same strain that were killed on the roads. Now, I'll come first of all to the kind of the advanced analytics before I get on to actually doing more systematic samplings. But there are now tools available which allow us to infer directionality within these phylogenetic family tree data. And the type of method is called a Bayesian structured coalescent, which sounds more intimidating than it is. Basically what it does is, let's go back to the tree we were looking at earlier, the same sort of one. And a Bayesian structured coalescent allows you probabilistically to figure out what the most likely host species was of these ancestor bacteria that we didn't sample. So there are mathematical tools that can infer that once they have the full tree that you have from an area. So for instance, this particular ancestor may have been in a badger. This particular bacteria may have been a cow. This one might have been in a badger. And once you have that information, you can then start to work out, well, who's transmitting to whom? And in this case, if you thought there's a high probability that this ancestor was a badger, then that becomes a badger to cattle transmission. And that's a badger to badger transmission. And so on across the whole tree. So you can quickly build up a picture of the transmission dynamics using these structured coalescent tools. And what they also give you is an insight into the diversity of the bacterial population in cattle and in badgers, which is useful, and I'll come back to that later. So then moving on to this, taking things this step further, we now have the analytical tools, and now it's about getting really good systematic samplings, particularly of badgers. And it's much simpler to get samplings of cattle purely because we have national schemes which are sampling cattle abattoirs, detecting their bacteria, growing them. And that's not really been the case for badgers except in exceptional circumstances. So the first place that a systematic sampling and really good analysis was done was this area of Gloucestershire called Worchester Park. It's an absolutely beautiful area. It's a gorge with a forest and a lake, surrounded by several cattle herds, but the actual park itself is really prime badger territory. It's been studied since the 1970s. It's an undisturbed site, so the problem with perturbation is unlikely to have occurred here. It's only about 11 kilometers squared, so it's about a tenth the size of the TVR area. But it also has one of the highest density populations of badgers in these islands. I mean, we're talking 30 to 40 badgers per kilometer squared at the time this study was done. So from this area, our colleagues and ourselves were able to get 257 cattle and badger M. bovis isolates over a 15-year period. We were able to then whole genome sequence them and then analyze them using the tools that I've mentioned, both bioinformatics and then this structured coalescent methods. And what we found, this is the family tree. It's been circularized just to make it easier to see. And what we see from looking at that is that there are these huge areas of the tree which are occupied only by badgers. So whole areas of whole branches, sub-branches, which are just dedicated to the one host species. And then further around, we can see the blue triangles here are cows, the red dots are badgers. We see that there's other sub-branches which have both species in them. And when we do the analytical technique of the structured coalescent, we can then start to figure out the directionality of infection in this system and see what it's like. This work was just published last December. And what we find from that work is that if we look at within species transmission, so we're really talking about cattle to cattle and badger to badger transmission here, we find that those occur twice as commonly as between species transmissions. Now there's a clear role for badgers in the Worchester Park system. But from that data, you could suggest that cattle are still slightly at greater risk from other cattle. But one of the really most important findings, I think, from this area is that when we do look at between species transmissions on their own, we find that badger cattle transmissions in Worchester Park occur 10 times more commonly than the reverse, cattle to badger. So the badger is really heavily involved in seeding infection into cattle here. But we also can't neglect the fact that there's within species transmission dynamics, which seem still to be the most important within the area. So Worcester was a really, really good chance looking at the systematically sampled badger population to get a really good data set together. And the TVR site is another golden-edged opportunity. I mean, when Fraser was coming up with this idea of doing this, we were rubbing our hands with glee, thinking, well, that's a perfect place to try this. And we were very, very lucky to be invited to do this as part of TVR, because I think we benefited probably from one of the best sampled areas in Britain and Ireland for doing this work. So kudos to Fraser for pushing this through, because I don't think we would have had the opportunity to do it as well as this otherwise. And also to the team who were out sampling badgers, because they did an extremely good job of getting us a well-sampled, systematically sampled badger population to look at. So from that area, we were able to get 619 M. bovis genomes, which we sequenced, some of them in-house, some of them we contracted out to another company. All of them were of exceptionally good quality. And then we were able to set up our own in-house bioinformatics system, which did an awful lot of the kind of the high-performance computing, which would enable us to do the transmission dynamics analyses later. In total, we had something like 218 billion base pairs of genome data, which is about 70 human genomes worth, if you want a yardstick to measure it against. Of those genomes, 119 were badger isolates, which were collected between 1999 and 2017. Although, cautionary here, we had multiple samples from the same animal for this. We also had 358 cattle isolates from within the TVR zone, collected over 1986 to 2016. So, as well as taking the samples collected in the trial, we were able to go back into AFBI has a really exceptional strain archive going back to the 80s and 90s, which we could go in and lift samples from this same area. We also had 75 cattle isolates from, there's the 2 kilometre buffer area around the TVR zone that Fraser spoke about earlier. And then we added in another 31 just for our own QA purposes. And here's the tree of the TVR area. So again, same color scheme, badgers are red dots here, bovines are blue. And this is a zoom out of everything that's in the TVR area. And the thing that becomes most apparent is that we have 8 different strain families, as Robin's kind of alluded to already. We see 8 different strain families in this area. And what we can see here is that the whole genome sequencing data is really highly correlated with the MLVA typing, the strain typing data that Robin was talking about. So you define a group with the strain type, and then the genome sequencing data just explodes it out into much more variation. And from the 8 families that we can see that were in the TVR area, we reckon that actually 7 of them were just passing through. They were ones which had probably come from outside, probably via cattle move. Some of these strains reside in other parts of Northern Ireland where they have a home range. And they weren't the ones that we really wanted to focus on, because the one that was causing the problem in the TVR area was the strain type 6, which is this whole, I've bounded it here with this green circle to show you just the extent of it. So the majority of our samples, and the ones which are most likely to tell us something interesting about transmission dynamics, are in that particular part of the tree. So moving on. This is then a zoom in on that part of the tree, which is just the endemic strain. This is strain 6. And one of the things that really pops out from your eye here, again, the badger samples are red dots, the bovines are blue ones. And the thing that really jumps out is that, say, unlike the Worchester Park area, we don't have single sub-branches which are dominated by a single host species. We find that both host species are peppered all the way around the tree. And that's telling us something that there's ongoing transmission in both directions. This is a shared epidemic, bovine TB. In terms then of what we find transmission dynamics wise, so we can really only comment at the minute. We're still in the depths of analysis with this. So the within species transmissions we haven't looked at in as much detail, but we're getting around to it now. Between species transmissions, very interestingly, we're finding that actually cattle to badger transmission is occurring 3 times more commonly than badger to cattle in the TVR system. And this is very much the inverse of what we saw in Worchester Park. Worchester was 10 to 1, if you remember, going the other way. So TVR area at the minute, from what we can see, there seems to be a lot of cattle-driven infection going on. But that's not to say that badgers aren't involved. They are involved. It's just to a lesser degree. So it's a shared epidemic. The interesting thing, as I say, contrasting this with Worchester Park, one of the conclusions you sort of have to come to is that transmission dynamics may well be context dependent. So depending on which part of the landscape you look at, you might find something different. Now, there could be good reasons for that. We can speculate what they are. We can't confirm what they are. One of the things that might be affecting this is that Worchester Park has such a large density of badgers there during the period of the study. As I said, 30 to 40 badgers per kilometre squared. If you contrast that to what Maria was finding earlier, about maybe 5.5 badgers per kilometre squared, it's an obviously large disparity there. And also, Northern Ireland would typically have some of the highest cattle densities in Western Europe. I think it's County Down could be something like 117 cattle per kilometre squared. So those 2 factors could affect the dynamics quite a bit here. The other interesting thing, which Fraser alluded to the fact, as did Shane, that we had taken badger genetic samples, and we've been looking at the badger population genetic structure in the area just as a kind of a measure of their ecology. And we find that, as you would expect with a philopatric or territorial animal, closely related badgers tend to be closer together in the landscape, and the further you move apart, the more likely you are to be less related. So that structure stays the same through all of the TVR trial. And one of the things falling out from our genome sequencing data is that, well, if badgers are playing a big role in the TVR area, you might expect that more closely related badgers also have closely related M. bovis genomes, and that doesn't appear to be the case. So again, it's one more thing that's suggesting that badger involvement is still happening, but it might not be the predominant driver in the TVR area. One other thing to be aware of, and this came out of RBCT work, so this is a bit speculative, but Crystal Donnelly, who's an exceptionally good statistician who looked at the RBCT work in England, had suggested that badgers could be responsible for a relatively small number of infection seeding events into cattle herds, maybe about 5%, but then that within those herds into which they had seeded infection, cattle-to-cattle transmission amplified the effect. And then it becomes a philosophical issue of, well, where's the best place to intervene there? So there's, I think probably our phylogenetic methods using genetics are probably unable to really pick up on those amplification events. So it's something to bear in mind that we are really looking at seeding events and maybe not capturing everything that's happening. I'll quickly rush through this. This is an area in Cumbria, in the low risk area of Britain here in the northwest, so the Lake District. Typically hasn't had a problem with TB. Recently, back in November 2014, there was an index case of infection found here. 27 breakdowns on 22 farms, and also dead badgers found. And all of these animals shared the same strain type, the MLVA, but it was not an English strain. It was actually one of ours. And we reckon this had been imported from ourselves into Cumbria by a cattle move and had then found its way into the badger population. And when we did genome sequencing on this, it actually links back to the TVR area. So its most closely related isolate is from the TVR area. And it actually looks at the transmission dynamics within that area in Cumbria, which are going to be the subject of a paper hopefully very soon, are very similar to TVR. So it seems to be a primarily cattle-driven disease. So I'll finish up. Key points. Exploiting pathogen genetics allows us to do things that years back we wouldn't have thought were possible. We can actually start to look at transmission dynamics to inform policy. And it's going to revolutionize the way we do all types of kind of bacterial transmission work in the future, but it's particularly good for TB. And we're going to be hoping to apply this maybe to other areas to contribute to improved control of disease in animals. And finally, just to everyone who helped with this, it's a huge team. We kind of just did this final bit of it, but we wouldn't have had any of this without the exceptionally good sampling and other support as well that went on. So thank you.
Okay, thanks, Adrian. We'll now pass on to our next speaker, who on the agenda is Emily Coursier, but this talk is going to be presented by Liam Doyle. So over to you, Liam.
Okay. My name is Liam Doyle, and I'm part of the Veterinary Epidemiology Unit, and I'm going to be presenting this talk today for Emily. So I'll try and get through it here and try and get the main points put across, as Emily's not available.
Sorry, Liam, could you put onto presentation style so get full screen? Sorry, just a screen show sort of thing, but I don't know if you can see it.
If you swap the view and slideshow there, just Liam. So just where you have click display settings, if you click swap presenter view and slide show, yeah.
Is that it now?
Yeah, that's it.
Okay. Just so as said there, I'm doing this presentation for Emily Coursier, and it's on the subject of the actual tests that's been carried out. Now, so if you actually take the whole subject that we're talking about here, TVR, obviously TVR is test, vaccinate, and release project. Big part of it is the test, the actual testing of it, and that's what I'm trying to focus in on today, is the actual testing aspect of it, and especially the actual testing aspect of it that I suppose we're most interested in here is the actual application of the DPP, the dual path platform test, which Shane has shown earlier. It's the application of this test pen side in the field, and how that how that test functions. So this most of what I'm talking about here is actually it's based on work and results that have been generated by a submitted paper from Mark Arnold of APHA. So what I'm presenting here is in that actual submitted paper. Now, the actual talk that I'm going to go through here is divided into two parts. The first part is it's based on paper. It's sort of how the results are that are arrived at. So the results that we're trying to do here is we're trying to evaluate the actual tests that are used in the TVR project. How good are these tests? So when you evaluate tests, you talk about the characteristics of a test. So that test, how good or bad a test is. So the first part of the talk is just how did we get to a point where we can say how good or bad a test is, and specifically how good or bad the pen side DPP test is. And the other part of the second part of the talk that I'm going to go through is actual results generated out of that paper. And just to, you know, what actually did we see from all this information that was gathered. So just to get on now with the actual talk itself, so this, Shane went through this and showed you sort of the practical aspects of how the test worked, and the actual test itself it's single use trap side lateral flow test, as it's described. Single use is used once and thrown away after that. These tests were developed by a USA company. They were actually developed for use in deer, and I think maybe even elephants as well, but deer and elephants. And they actually are, within the data sheet, they would have been described, they used serum rather than whole blood. So they've also been used, as far as I can see, in a wide range of other animals too. They've been used in wild boar, South American camelids, and also in white-tailed deer in the US. There's a problem with mycobacterium, with bovine TB getting into that population. So the test, this DPP test, the test that's used in the field, it detects antibodies in the blood of the badger to Mycobacterium bovis. As Shane explained, there's 2 lines on this test, and it's just basically a test for 2 different aspects of M. bovis, line 1 and line 2. So that it tests for two different aspects of M. bovis. So as well as that, so just in terms of the tests that were carried out in the field, again, Shane mentions, but I maybe want to come at them from a slightly different angle. There was a range of tests carried out on these badgers that were caught. So if you actually look there, the actual tests that were delivered, they're described as both field tests and laboratory tests. And there's results from all of these tests. And if you look in the field, the type of things that were carried out, all badgers were tested in the field. There was 10 microliters of blood placed into that DPP test kit and tested in the field. So that was one test was carried out. Also from the badgers, blood was taken. And Shane showed you the different tubes and that those blood samples were put into. Those are brought back to the lab. There was also clinical samples taken in the field, and these comprised of tracheal aspirates, wound swabs, and oropharyngeal swabs. And then within the lab, you had other tests carried out. Now, within the lab, the DPP tests were repeated. Now, the DPP test in the lab was carried out in two different samples. One, it was repeated on whole blood in the lab, and it was also carried out on serum. So, in other words, within the lab, you were able to take and spin down these bloods and actually test serum. Plus, you were able to use whole blood to verify the actual field results as well. Also carried out to the lab, you had these. You had another test, just a different test. The interferon gamma test was also carried out in the lab. Again, as a complex test, it can only be carried out in the lab, but it's actually an important. It gives important extra information about the overall test. And the other tests that were carried out in the lab were culture of the clinical samples, such as the culture of the swabs and aspirates and wounds swabs, and also for any badgers that were removed, they were post-mortem. Now, I suppose the important thing, one of the reasons I'm talking about this whole list of tests is that you can see that the one we're really interested in here is the field test, the whole blood. How does a whole blood sample on the DPP test kit in the field work? But we'll take it. There's a whole range of actual samples taken here. Now, if you can imagine, every one of those tests tells you something about the state of the badger that you're testing. And ultimately, what you're trying to do is you're trying to say, in terms of diagnostic testing, what you're actually trying to figure out is that badger in front of you, is it infected or is it not? And here you have a whole range of tests. Now, what I want to show you in the next slide is just what do all those test results look like. I don't want to go into this in too much detail, but I want to sort of show how you get from all these tests to the characteristics of the test. So if you look here, this is just an example of 2015 and 2016 figures and the test results. What you can see here from these test results, you can actually see there that the majority, actually 82% of badgers tested negative for everything. And that's the top row in that table. Now, in terms of the DPP positive tests, you can see on the bottom 1, 2, whatever, six rows there, those are the DPP positive tests. And that was in 14% of cases that those DPP tests were positive. But you can also see that just because you get a positive DPP test, you could, if you actually go across, you can see some of them will give you a negative interferon gamma test, some will give you a positive. Some of those will actually then give you positive clinical tests, negative clinical tests. And then if you go carry it on to post-mortem, you can see that there's various, you can see within all of these tests, there's a range of combinations and permutations of these tests. Now, what I'm trying to get at here is that what we're trying to do here is take all these tests and build them up into a picture which shows us what the test characteristics are for each of these tests. In other words, how good are these tests? How well do they function? So I don't want to go into any detail on the actual mechanisms of how to do this, but just to say that there are statistical methods for these known as Bayesian frameworks, which take all these various test results and put them together. And from that, you actually can determine how good or bad tests are. How good or bad tests are known as the characteristics of the test. Now, characteristics of a test, they're just, all that these are is that they're telling you how well that test performs. And it's the same process in any aspect of life. Like, that you look at, if you're buying a car or anything, there's certain characteristics of it that you look at that tell you how good or how bad it is, or as to making a choice. This is the same with tests, and the terms that are used that to tell you how good or bad a test are the sensitivity and the specificity of the test. Now, to keep just sensitivity of the test, it's all about how good is that test at picking out infected animals. Okay, so you want a test to be very good at picking out, if an animal is infected, you want that test to tell you that animal is infected. And that's the sensitivity of the test. But equally, the specificity of a test is telling you about how good that test is at discriminating non-infected animals. So for a test to be very good, you want it to pick out infected animals, but you also want it to pick out uninfected animals. But you don't want a test returning you a pile of false positives, because it becomes useless if you've got an awful lot of false positives. So the sensitivity and specificity of the test tell you how good that test is. So as I said earlier, all the results that I'm going to present now in the next part of the presentation are based on this paper, submitted paper where Mark Arnold has done the analysis in it. Now, Fraser had mentioned this earlier. There was this analysis had to be split between years 1 and 4 in the final year, because the Badger BCG was only available for the first 3 years, and then in the last 2 years they had to revert to this other BCG Sophia. So in terms of the analysis, these two, when you use the different vaccinations, they're obviously going to have an effect on the characteristics of the tests. So there's no point in analysing it over the whole 5 years to look at the characteristics of the test, because you're using different vaccine product in those two parts of the study. So the results. Results from the actual project. Just a few of the headline things that we that the results actually show. And this first one is a very important result and a very important take-home message out of it. The results are not affected by use in the field. We didn't see that if you test whole blood in the field versus testing the whole blood in the lab, you got any different results, which is a very good result and shows that the field testing is really as good as taken to the lab. The best overall test characteristics, in other words, the best sensitivity specificity combination that we observed during the actual project was to use the Dpp test. That's the test Shane showed you in the field, using whole blood and using it pen side. In other words, the best test characteristics we observed were to take the test and use it beside the badger in the field with whole blood. Now, the other thing that we also observed from the results were that, and Shane showed you this as well, that within the actual Dpp test, there's the line 1 and the line 2, and you can interpret these tests. No, you could actually interpret the test using line 1 only, using line 2 only, or a combination of line 1 and 2 to see, to remove badgers. But the results show that if you forget about line 2 and use line 1 only as your result, to determine whether you should remove or leave a badger, if you use line 1 only, that gives you the best results. And within the Arnold paper, it's all based on results using line 1 only. The other result we saw within was that there was good reproducibility for line 1 between different people reading the test. So in other words, this is good news for field results as well, that different people reading that test in the field, it didn't seem to make a pile of difference. The decision one person made was as good as the next. And a final headline result from it was that when we compared BCG Sophia to the Badger BCG, we found that the BCG Sophia had a much greater negative effect on the performance of the Dpp serum test. Now, in other words, the blood samples from the field, some of them, they were all taken back to the lab and they were spun down and they were checked with serum. But it appears that with BCG Sophia, that it seemed to affect the performance of the Dpp serum test negatively. However, the good news was that it did not appear to have a huge effect on the actual whole blood test. So that's a good news point from it, that it's only the serum test that the BCG Sophia vaccine appears to really hit. So the actual results from the paper. If you look at this table, now this table actually gives you the sensitivity and the specificity for this whole range of tests. So these are the figures that are telling you about how good your test is. Now, the one I suppose you want to look at there is the actual second one down, Dpp whole blood sensitivity. If you can see, the sensitivity of the Dpp whole blood test there is 0.69, and there's a credible interval range given with it as well. But so, in other words, what that test telling you is you have a sensitivity of approximately 70% there. So if you present that test with 10 infected animals, it'll pull out 7. So, and if you compare that to the other two tests either side of it, the actual Dpp serum test and the IFNG blood test, the two laboratory-based tests, the Dpp whole blood of the field appears to act about, it's about 10% better than either of those in terms of sensitivity. And the Dpp serum and the IFNG lab-based tests appear to be fairly similar in terms of their characteristics. In terms of specificity, all these tests performed quite well. They're all in the high 90s. So they're not actually returning you a pile of false positives either, which is reassuring in terms of the tests. So the bottom 3 rows in the table show you what happens whenever you apply these tests with BCG Sophia. Now, I suppose there is a point to be made. The BCG Sophia was only used at the latter part of the project. So if you actually look at the intervals, they're wider. We're possibly less certain just about the information that's, that we have here because we have less data. But if you actually look at the second last row, DPP serum for BCG Sophia, and go over to the specificity, you can see it has dropped to 0.81 now, which in reality is going to if you use that test, that situation, you're removing a lot of false positives. So you're removing a lot of badgers with a test positive that are not infected. So that is actually it's a sort of a downfall on that test in that situation. Now, I suppose from this from this paper as well, the other information is returned. Now we have information on sensitivity and specificity of the tests. We can look at other things as well, and in that paper, the other thing that was looked at was how could all of this knowledge, the sensitivity, specificity, along with the test results on badgers, how could that be modelled into some type of representation of how the actual prevalence of disease is developing within the badger population over the period of time of the study? And if you look here, this is just raw data, the raw results that Fraser's generated, which show you the actual proportion of animals which are positive to the test over the years of the project. So in other words, this is just showing you DPP positive test results over the time period of the project. And you can see there that just looking at that, what is an apparent prevalence, because you're just looking at test results, it possibly looks like there is reduction in it, although you have to look at the actual confidence intervals there. They are wide and there is overlap in them. Now, within the paper by Mark Arnott, there is an actual a model developed where the actual sensitivity, specificity, and the parameters of the test are taken into account, and he has looked at what actually is happening, actual infection prevalence over the period of the study in terms of what's happening in the actual badgers. And this model, which is shown, is the best sort of fit of what is happening within the badger population. Again, before I say anything much about it, you can see that there are lines on the top of the bars, or which show again there's wide confidence intervals and or credible intervals in this data. But what it's showing from what we can determine from the data is that there is a reduction year on year in the actual infection prevalence in badgers over the years of the project. And in terms of the model, what does it show? If you go by what it's showing, it's actually showing that from year 1 to year 5, there's a reduction in the actual infection prevalence in badgers from 14% down to 2%. But again, this is a model, and as you know, all models come with health warnings, and there's probably more data needed to back this up. But it is a very encouraging result. It does show that there is a tendency for a reduction in prevalence of bovine TB in the badger population over the years of the project. So just to summarise what the talk there has been about, what we've learned from this is that the DPP test, this pen side test, it's suitable for use in the field, and it's suitable for use with whole blood. It is suitable for use reading line 1 only, and that's probably the best way to go. Don't bother with the line 2 in it or any combinations of line 1 and line 2. And there's also tentative evidence there that there's a reduction in infection prevalence in the badger population over the period, and also that also the effect of using BCG Sophia. It seems to mess up the DPP test in terms of using serum, and so that's the talk related. Thank you.
Thanks very much, Liam. We're running slightly over time, so I'll just pass straight over now to Fraser, who'll deliver Emma Campbell's talk on badger and cattle interactions and cattle grazing patterns.
Better if I unmute. Thanks very much, Liam, for standing in at short notice to give that and did it far more comprehensively than I would. I'd just like to give a brief overview of some work Emma Campbell has done on a PhD within the TVR area. Unfortunately, Emma couldn't do it herself, but we'll maybe get her back for some of our lunchtime seminars later on in the year. This was originally part of the badger-cattle interactions that was the initial TVR proposal, but had to be cut down with the ministerial directive. So Emma had a few farms she took on board within the TVR area, and then she ended up with different number of farms for different studies, but within the one she was looking at indirect contact between badgers and cattle, she had 34 farms in which she placed motion activated cameras. You'll see a picture of one there on 6 sites across each farm for 2 separate weeks over the year, 1 week in the winter spring period and 1 in the summer autumn. She put them in 3 badger locations, as they were called, at a set, a latrine and at a badger run, and at 3 cattle locations, which are a cattle shed, a feed store and a water trough in the field. So over the 34 farms, poor Emma had an awful lot of hours of video to watch to distill down the actual results. There's lots of animals recorded on the videos, but about 6.5% were badger detections and about 7.2% were cattle detections. Interestingly, of the 34 farms, she never found any badgers entering into the cattle sheds or the feed stores. On average, she had 8.5 badger detections per farm per week, and 0.8 detections per week of badgers near water troughs. Now, they were caught on camera, but they very rarely used or observed drinking from the water troughs. It's about once every 87 days. Also, when she analysed the results, we found that cattle are 3 times more likely to visit a badger location. On average, this happened every 3 days. And I think there were 6 occasions in which they were observed sniffing down the sets of entrances. As we know, cattle are very inquisitive creatures and they'll explore anything unusual within the environment. And when we look at, there's obvious evidence of indirect contact between these species at the different sites, and the time interval between the cattle visiting and badger visits to each site were, within a few hours of each other, usually less than 29 hours, which would suggest that the M. bovis bacteria can survive well over that length of time. So there's potential for transfer. I'll say that this is available in Research and Veterinary Science on Ecology and Evolution, and the paper is readily available. I'm just rushing through it. Let's say we'll get Emma back to do it in a more focused approach. So, I mean, the department's been telling farmers for ages to fence off badger sets, and really to stop access to cattle to these sets, to stop this sort of activity here. See, here's a badger set here it's quite a rudimentary but effective temporary fence to keep the cat away from this badger set, which is located here, and this is the area, the big spoil heap where the badgers excavated the soil. She also, about 2016, monitored 18 farms in which she looked at the grazing patterns within these farms. So the farmers were very kindly agreed to record where each of the cattle groups within the farm were placed and which fields they were placed at what time. And this occurred, they took recordings from May to November, using the farm maps to identify the fields and recording these in books. And from the daily inspections, they also recorded whether there's neighbours' cattle in adjacent fields. We also had details on the farm size, and presence of badger sets of latrines, and we also had from these 18 farms the previous TB histories from these farms. So the monitoring involved nearly 700 fields over in excess of 200 days, and of these fields, about 60% of them were grazed during this period. The average grazing time in each field is about 63 days, and these are just, these are the average size in fields. Obviously, dairy cattle herd size was a lot bigger, but they all had quite a big, there's quite a high prevalence of TB in the area, with nearly 50% of herds having TB in the last 3 years. When we look at exposure, only 3% of the fields actually had a badger set recorded with them, and 64% of these fields were grazed. And when we look at the cohorts within the herds, cows and calves spent most time in fields with badger sets, and dairy cattle spent more time in fields with badger sets. And when we look at compare this with the TB histories, they were significantly related with the time spent grazing fields with sets. On the latrine side, again, about 3% had latrines. These did vary considerably from the ones with sets. And again, about 60% of these fields were grazed. And heifers and cows were most commonly observed in these fields with badger latrines. But we actually found a negative association between the history and grazing time for these fields with latrines for the 3 and 5 year history, but not in the last 12 months. So of the 18 herds, 2 had recorded no contact with other herds, neighbouring herds during the grazing season, but 90% did have contact. And the median grazing time spent next to neighbours was 18%, which is a lot higher than I would have expected. And the contact time varied with the life stages, but I won't go into that. Emma can present that when she comes over sometime. And we found no relationship between neighbouring herds, the time spent grazing next to them, and the area of fields, herd size, or TB herd history. But again, it was quite a small sample size. This paper is in Research and Veterinary Science if you want more detail on it. She has carried out other studies which she's writing up, looking at the cattle movement metrics within the farm, and also at the latency which badgers can start using a field after cattle have grazed it. And the platform of the TVR study has also presented for other other studies in badger ecology, for example, with the Queen's students putting accelerometers on badgers, which do very detailed recording of badger movements, and that's other work that's ongoing. So to summarise, cattle are more likely to visit badger locations, and there seems to be a herd history associated, TB history associated with grazing farms, fields with sets in them. So again, our advice stands of fencing off badger sets to prevent access. Badgers do infrequently visit water troughs, but perhaps farmers need to look at re-evaluating the positioning of water troughs in fields. And badgers were never seen to go into farmyards, but the other studies have shown there's quite a large variation, and some badgers do seem to get in the habit of visiting farmyards. So it's very, the assessment of risk from badgers really has to be on a farm-to-farm basis. And we saw 18% of time spent grazing next to neighbouring cattle. These are all points which maybe practical steps can be taken to mitigate against these. And sorry for this rush through, but just as by way of pictures saying a 1,000 words, here we have from our TVR study, if I can get it going. Here's a badger out one night, just marking its spoil heap. That's just outside the set. So it's done its business and disappears off. Again, this is the motion activated cameras in action. And then we see that this is the same camera. Where's me? There it is. So we see a few hours later this inquisitive beastie coming along. Notices the smell, and you can see by that sort of action it could quite easily aerosolize any infective material that's lying about. So, sorry for that quick rush through. Emma's work requires a lot more detailed, but as I say, she's working up other parts of our study, and we'll get her over sometime in the future to present some more of her stuff. So I think we're running behind time, so quickly hand over to Raymond to kick off the question and answer session.
Right, thanks for that, Fraser. Thanks for stepping in for Emma, and thanks for also making up a little bit of ground for us. We are running slightly over time, but I think you'll all agree that there was lots of good stuff there, and it was well worth hearing. So we still have time for a few questions, and we'll move into that now. I'm delighted to say that there have been a good selection of questions. Just to remind you again that today is all about TVR, what we did, how we did it, and what we found out. So the questions that we'll pick out to deal with will reflect that emphasis. The first question is actually one for me, and the question is, can I get a copy of the presentations? And the answer is yes. If you're a member of staff, these presentations will be published on the intranet site, and you'll be able to look at them there, as will any colleagues that weren't able to join us this morning. If you're an external invitee, if you get in touch with the Veterinary Service Animal Health Group Wildlife Unit at the email address shown, we'll get you a copy sent out to you. So moving straight on, the next question is one for Maria, and the question is, how did the number of sets actually found in the project compare with the number predicted by the previous Queen's University modelling work? So Maria, if you would like to respond to that.
Yeah, actually the Queen's modelling work was done in 2008, and there was a little bit of modelling work done before that in 1993, as far as I remember. And the badger population has stayed fairly stable over that time. And so the TVR sets that were found actually reflected that stability as well. So there's a good match between the two.
Thanks, Maria. Thanks for that. Our next question is one for jointly for Robin and Adrian. And the question is a very interesting one. What do we know about these badger strains? Is there such a thing as a good strain or a bad strain?
Of the pathogen, yeah. Yeah, I'm going to, Adrian can jump in, of course. The starting point for, I guess, our thinking on this is that it's biologically untenable that variation, genetic variation in the host, that's cattle, and in the pathogen doesn't have an impact on the outcome of infection. That's our default sort of starting point. But I'll qualify that by saying that I think in the TB system that we have in Northern Ireland, I think there's just so much noise in it that it's very hard to unpick the pathogen features. Yeah, we have had a sort of statistical look at whether particular strains are associated with large outbreaks as a proxy measure for transmissibility, and we didn't find anything significant. We did find some strains in the pathology abattoir data tended to be found in particular organ groups and carcass sites. An interesting observation. And we have recent data that suggests a slight strain effect on the size of the reaction in the skin test, but there's such variation and such noise in it that it's very difficult. I wouldn't want to overreach that one. But you would imagine the TB is a clonal pathogen, so it will respond, it will evolve, and it will respond by a mixture of selection and genetic drift. So there's an interesting suggestion that maybe the strong application of test on slaughter in the last decades has selected strains that don't react or react less in the tuberculin test. And we've actually been able to get isolates that predate the tuberculin test, and some of them have been sent to other countries around the world. And our feeling is that they're still, the same clone is represented there. So the feeling is that what we have at the minute is just the lucky clone that got through the bottleneck that was applied by the tuberculin test. That there's nothing all that's peculiar about it. It still can infect other hosts, and it still transmits. It's still adapted to cattle. That's kind of where we stand. I think just take home is I think there's a lot of noise there. It's a really interesting question and a potential career break. Do these strains have different phenotypes that are relevant in the program?
Thanks, Robin. Adrian, anything you'd like to add to that?
No, I think it's I think the null hypothesis, which I think I would probably be close to thinking is probably most likely true, is that there's not much difference. I think the particular lineage of M. bovis we have, I reckon, is fairly bottlenecked, but there's not an awful lot of diversity in it. So I think looking within region won't be unlikely to find kind of specific traits of strains that are having a massive effect. If we were able to compare against lineages from other countries, we might find something where that's certainly been found for M. tuberculosis in humans. That like there's a thing called the Beijing strain, which is a hugely more virulent strain than some of the other ones around the world. So I think if we cast the net further, we might stand a chance of finding something. But if my guess would be there's nothing meaningful there, but it probably wouldn't do any harm to look a bit harder to absolutely rule that out.
Right. Thanks to you both.
Sorry, Raymond, just Fraser here.
Yes, Fraser.
Epidemiologists usually avoid anecdotes, but it was interesting TVR how there was one badger we caught for 4 years and consistently got isolation of a Pacific, unusual strain of Mycobacterium bovis from it for the 4 years, but it always proved negative to the DPP test. But it was just an interesting observation. It's frustrating how we knew that was positive, but we couldn't remove it because it was negative to the DPP test.
Okay, thanks for that. We'll move on to our next question, and this time it's one for Shane. You saw earlier on this morning that GPS collars were attached to some of the badgers, and we've had a question in, Shane, about the recovery of those collars. If you'd like to comment on that, please.
Yeah, Raymond, we had good recovery rates in that we recovered most of the collars that were put on, and our recovery rates compared quite favourably to those that were published in any data that was available from other studies. We were able to monitor the activity via the online viewers when the collars were working. The collars worked for approximately 6 months, depending on how many readings they were taken at night. We also monitored RTAs in the area. Farmers were aware that badgers may have been collared in their areas, and farmers did find some. You could also detect some of the collars with a UVF radio detector. We tried that on a few occasions, and then those that we didn't recover during our initial sweep through the area, we would deploy infrared cameras and see if we can detect the animals on the cameras, and then we would also put traps back in to try and trap those animals again and to recover the collars. The collars in general, they were well tolerated. Some badgers received a collar year on year. We did make some refinements over the duration of the project around the weight of the collar. Initially, there was a drop-off mechanism on them, which we removed after year 1, and then also the strap design changed in conjunction with feedback that we give to the TELUS company that produced the collars for us. So yeah, no, we had a good success at getting them back.
Right, thanks very much, Shane. And we'll just take one final question, and it's back to the guys in the lab again. The question is, BCG is a live vaccine. Did we isolate BCG from either badgers or cattle within the TVR area?
Okay, yeah, well, I mean, you're right. BCG is at least some component of BCG is live, and it's derived from a French bovis strain originally. And we did isolate it from 4 roadkill badgers and 2 TVR badgers, if I remember correctly. But it was isolated in culture from the lymph nodes that would have been draining the site of injections. It wasn't a full-on excretion. It was we recovered it from the lymph nodes, and the genetic tools that we talked about this morning they're good enough that we could distinguish between BCG Danish and BCG Sophia. So that's quite a useful tool in the box. But I mean, important point, Raymond, we did not isolate BCG from cattle, which is good.
Absolutely, yes. Thanks for that, Robin. Right, everybody, I think you'll all agree that we've got our money's worth out of our speakers and that we should let them away to have something to eat. So I'll just wind up the session now. We haven't been able to deal with all the questions that came in. So if you'd like to follow up on any of today's presentations, please drop an email to the Wildlife Unit. If you can, include in the title of your email TVR Day and to whom you want to address your question, and that will help us handle them at this end. In closing, I'd like to just thank all our speakers again. They all have been intimately involved in the project, and I hope that you found their insight and the depth of knowledge they have to be useful. I'd also like to thank Carl Moyne, who has been working away furiously in the background, trying to keep the show on the road in a coherent way. And I would include in that muting my own microphone when I forgot to. So thanks very much, Carl. And I hope you learned a lot about TVR as well. I would like to just remind our own frontline staff listening in of your key role in delivering a consistent and accurate message to our farming community on TB. It is a controversial issue and it is a difficult issue. So I hope today will have helped you to do that. As I say, the talks will shortly be placed on the intranet, so feel free to view them again at your leisure, run back over them again, and also encourage your work colleagues to do likewise. Finally, I'd like to thank our invitees for your interest in our event today. I'm pleased to say that around about 100 people have listened in live, and I think this is a testament to the general interest in this work and the quality of the research which the team carried out. So, as I say, thank you, and I hope you enjoy the rest of your day.